Hello everyone. Today we are going to start with the components of the electron transport chain. Electron transport chain consists of 4 enzyme complexes and 2 free electron carriers. So the electron has to flow through a series of complexes or components, and finally it will combine with oxygen, and there is one more complex 5, which then produces ATP. So regarding that we are going to study the components of the electron transport chain. The components consist of 4 components or 4 complexes. Complex 1. Complex 1 is NADH ubiquinone oxidoreductase. This is complex 1. Now, complex 2. Complex 2 is succinate dehydrogenase enzyme. Complex 3. Ubiquinone cytochrome oxidoreductase. And complex 4 is cytochrome oxidase. So there 2 electron carriers are coenzyme Q and cytochrome C. There are 2 electron carriers and 4 complexes. Along with this complex, one more complex, complex 5, which is an ATP synthetase. It is nothing but it helps in the synthesis of ATP, or the enzyme ATP synthetase is present. The complex 1 to 4 are the carriers of electrons, and the complex 5 is responsible for ATP synthesis. This is a slide which shows about the complexes. We have talked about 5 complexes are there: complex 1, complex 2, complex 3, complex 4 and complex 5. So the electron will flow from complex 1 to complex 3, complex 4, and finally it reaches complex 5 where ATP synthesis takes place. Or the electron will flow from complex 2, 3, 4 and 5. Okay, if the electron is flowing from 1, it bypasses 2, it will go to 3, 4. If it is flowing from 2, it will flow through 2, 3 and 4. It doesn't go 1, 2, 3. It goes as 1, 3 or 2, 3 and 4. The enzyme complexes and mobile carriers are collectively involved in the transport of electrons. Finally, they combine with oxygen to produce water. The largest production of oxygen supplied to the body is utilized by mitochondria for the operation of electron transport. We have to study about the different complexes. What are the role of these complexes? What it consists of? The complex 1 consists of 2 coenzymes NAD plus and NADP plus. NAD plus is more actively involved in electron transport chain. This is tightly bound to the inner membrane of mitochondria. NAD plus is reduced to NADH and H plus by the dehydrogenase enzyme. How? From the substrate, 2 hydrogens will be removed. Okay, the substrates can be pyruvate, glycerol 3-phosphate. The 2 hydrogens will be removed. It will be accepted by NAD plus, and the NAD plus will get converted or get reduced to NADH and H plus. NADPH is also utilized when during anabolic reactions. Which type of reactions? Anabolic reactions. During fatty acid synthesis and cholesterol synthesis, NADPH is utilized. If the substrate is pyruvate glycerol, NAD is involved, and when the substrate is fatty acid synthesis, cholesterol synthesis, NADPH is in. Here you can see from the substrate, XH2 means the substrate. XH2 means the substrate. From the substrate, 2 hydrogens are removed, H2, and the substrate will become X. That 2 hydrogens will be accepted by the coenzyme NADP+, and it will get reduced to NADH. If NADP plus is accepting, it becomes NADPH plus H plus. You see the complex 1. Complex 1 consists of the NADH coenzyme Q, or NADH dehydrogenase. This is complex 1. The flavoproteins. What are flavoproteins? Flavoproteins are nothing but the enzyme NADH dehydrogenase is a flavoprotein with FMN as the prosthetic group. What is prosthetic group? The group which is present along with the enzyme, the non-protein part of the enzyme. The enzyme consists of protein part and non-protein part. Protein part is the enzyme, non-protein part is also present, which is called as prosthetic group. What happens to this? This FMN is a prosthetic group which will accept the 2 electrons from NADH and H plus, and it becomes FMNH2. Becomes FMN. You see here, from the substrate can be glycerol, 3-phosphate, the 2 hydrogen atoms are removed and it is accepted by NAD, it becomes NADH and H plus, and this is accepted by the prosthetic group FMN. FMN becomes FMNH2, and finally it is given to FES. That also we will see in the later. Now this NADH plus H plus FMN becomes NAD plus, because NADH and H plus, the hydrogens are given to FMN, and FMN becomes FMNH2. In this, the 4 protons are pumped out from the. We will talk about complex 2. Complex 2 is the enzyme succinate-dehydrogenase or Succinate-CoQ-Reductase. CoQ means coenzyme Q reductase. The electrons from this succinate-dehydrogenase enzyme, it will get the electrons from FADH2 will enter the electron transport chain, and then it will be given to coenzyme Q. The succinate-dehydrogenase is an enzyme which is found in the inner membrane of the mitochondria. It is a flavoprotein with FAD as the coenzyme. The 3 major enzyme systems that transfer the electrons to ubiquinone is succinate-dehydrogenase, fatty acyl-CoA dehydrogenase, mitochondrial glycerol phosphate dehydrogenase. These are the 3 enzymes involved. Now we can see that the complex 2 succinate-CoQ, that is coenzyme Q reductase. From the succinate, the electrons will be given to FAD. FAD will become FADH2, and from there FADH2, the electrons will be given to FES. What is FES? We will see in the next slide. So, the electron transport chain will start with succinate or will start with complex 2 when the substrate is succinate. If the substrate is glycerol or pyruvate, it starts with complex 1. This is the coenzyme, flavin coenzyme. They are showing FMN becoming FMNH2. Yes. What are these iron-sulfur centers or iron-sulfur clusters? So we have seen in the previous complex 1. Finally, the electron reaches FES. In the complex 2, after FMN, it is reaching FES. What is this FES? FES is iron-sulfur centers or are prosthetic groups containing 1 to 4 iron atoms. This iron-sulfur proteins exist in oxidized or reduced state. Oxidized state means Fe plus 3 and reduced state means Fe plus 2. The iron-sulfur center transfers only one electron, even though 2 or 3 electrons are reaching at that position, but it will transfer only one electron. Then, this FES participates in the transfer of electrons from FMN to coenzyme Q in the case of complex 2. Yeah. Other FES proteins associated with cytochrome B, cytochrome C1 participates in the transfer of electrons. In other places, in the next complexes also we can see where cytochrome B and cytochrome C1 is also associated with FES, which helps in the transport of coenzyme Q. Coenzyme Q is called as ubiquinone. It is a quinone derivative with isoprenoid side chain. Mammalian tissue possesses quinone with 10 isoprenoid unit, which is known as Q10, coenzyme Q10. This ubiquinone, that is Q, is reduced to semi-quinone QH and finally to quinol QH2. The next slide will show the structures of QH10, QH2 and QH semiquinone. This ubiquinone accepts a pair of electrons from complex 1 NADH or FADH2 or from complex 2, and then it will be given to the next complex 3. So this ubiquinone or Q10 or coenzyme Q acts as a, in this slide we can see coenzyme Q, ubiquinone Q. It is fully oxidized. How it is getting converted into fully reduced QH2, that is ubiquinol. Ubiquinone becoming semiquinone QH and finally ubiquinol QH. Cytochrome reductase or complex 3. This is a cluster of iron-sulfur proteins, cytochrome B, cytochrome C1, and both contains heme prosthetic group. Cytochromes are conjugated proteins. So since it contains heme, it consists of porphyrin ring with iron atom. Heme group of cytochrome differs from the heme of the hemoglobin. The ion of heme in cytochromes is alternatively oxidized and reduced. That is, it will itself get converted from Fe plus 3, Fe plus 2 and Fe plus 2 to Fe plus 3, which is essential for the transport of electrons in the electron transport chain. In this, the 4 electrons are pumped out. The electrons transported from coenzyme Q to cytochrome B, C1, C, A and A3. That means the electrons from the coenzyme Q will now pass through cytochrome B, C1, C, A and the property of reversible oxidation reduction of heme ion present in cytochromes allows them to function as an effective carrier in electron transport chain. So, why heme is used? FES, we have seen FES as a carrier. Why we are using that? Why it is used? Because it has the property of oxidation reduction. It itself will get converted Fe plus 3 to Fe plus 2, Fe plus 2 to Fe plus 3. So, it functions as an effective carrier of electrons in electron transport chain. Now, what is this cytochrome C? It is a small protein containing 104 amino acids and a heme group. It is loosely bound to the inner mitochondrial membrane and can easily be extracted. What is complex 4? Complex 4 is cytochrome oxidase. It consists of cytochrome A and cytochrome A3. So complex 4 contains cytochrome A, cytochrome A3, which is the terminal component of electron transport chain. It is also tightly bound to the inner membrane of the mitochondria. Cytochrome oxidase is the only electron carrier, heme ion of which can directly react with molecular oxygen. It acts as a carrier, electron carrier. The heme ion helps. It also contains copper that undergoes oxidation reduction during the transport of electrons. The two protons are pumped out of the electron transport system. In the final stage of electron transport chain, the transported electrons, the free protons and the molecular oxygen combine to produce water. So in the final stage, as the electron from the substrate will pass through the complex 1, complex 3 and complex 4, and finally it will combine with oxygen to produce water. Yes, we will see now the whole electron transport chain, how it is working. So we have studied complex 1, complex 2, complex 3, complex 4. Complex 5 I have not explained. Okay, I will explain in the future slides. Now, if the substrate is pyruvate or glycerol 3-phosphate, it will give its electrons and it will be accepted by NAD plus and get converted into NADH and H plus. This NADH and H plus will give its electron to FMN and FMN will become FMNH2. This will be given to FES, which acts as a carrier. This is what happening in complex 1 by the help of enzyme NADH coenzyme Q-reductase. This FES will give its electrons to coenzyme Q. Coenzyme Q will give to complex 3. Complex 3 consists of coenzyme Q cytochrome C reductase, which again consists of cytochrome B, FES, cytochrome C1, cytochrome C. Through this, the electrons will flow and will reach complex 4. Complex 4 is cytochrome oxidase, which consists of cytochrome A, cytochrome A3. The electrons from this complex 4 will combine with oxygen and will form water. Okay, this is how from the complex 1, 3, 4 the electron is flowing. If the substrate is succinate, then complex 2 will take part in the electron transport chain. Complex 1 does not take part. The complex consists of succinate. From succinate, the electrons will be given to FAD. FAD will accept. FAD will get converted into FADH2. FADH2 will give its electrons to FES. FES will give to coenzyme Q. Coenzyme Q to complex 3, 4, and finally the electron will combine with oxygen and form water. These slides are telling about the source of electrons, electron donors from complex 1. What are the sources of electrons? Electrons NADH and FADH2. NADH is produced from which and all reactions, like from PDH complex, from alpha ketoglutarate, from isocitrate dehydrogenase, malate dehydrogenase, hydroacyl coenzyme A dehydrogenase. What are the sources of FADH2? Flavoprotein, it is produced by the following reactions: succinate dehydrogenase, glycerol 3-phosphodehydrogenase, fatty acyl coenzyme A dehydrogenase, and all these from complex 1. Or we are going to see about inhibitors of electron transport chain. The inhibitors bind to one of the component of the electron transport chain, either complex 1 or complex 2 or complex 3, complex 4. Anywhere it can block. So when it binds, it blocks the transport of electrons. Then, what are the causes of it? It causes the accumulation of the reduced component before the inhibitor blockage, and after that, after the blockage, the oxidized components will be accumulated. So before the inhibition, reduced components will be accumulating. After the inhibition, oxidized components will be accumulating. Now, what is the role of this ETC? What can happen? If the inhibitor binds to the electron transport chain, the synthesis of ATP does not take place, because the synthesis of ATP is taking place in complex 5. So, the synthesis of ATP is dependent on electron transport chain. This slide will explain the ATP synthesis and the inhibitors. Okay, so the inhibitors, if the substances like amytol, rotenone, pyridine A, these substances are present, then they will inhibit at site 1 or complex 1. If antimycin, BAL, these substances are present, they inhibit the site between cytochrome B and cytochrome C1. Cyanide, sodium azide, carbon monoxide, they will inhibit the site between cytochrome B and cytochrome A3. So it is written ATP site 1, ATP site 2, ATP site 3. So these places ATP is produced. Yeah, if amytol, rotenone, pyridine is inhibiting, then the first site, the complex 1, ATP synthesis does not take place. The second, if antimycin A, BAL, if they are inhibiting, then at the second site doesn't take place, and cyanide at the third site. We can see the complex 1, yeah, NADH coenzyme Q. It is inhibited by rotenone. What is rotenone? It is a fish poison. Then amyltol, it is a barbiturate drug. Then antibiotic pyridine A. Complex 2 is inhibited by between cytochrome B and C1. Complex 3, it is inhibited by antimycin A, which is an antibiotic, BAL, which is an British antiviscite. Yeah, it is used as an antidote during the war gas. It is used as an antidote against the war gas naphthoquinone. These are important inhibitors between the site cytochrome B and C1. Cytochrome oxidase, complex 4. It is inhibited by carbon monoxide, cyanide, hydrogen sulfide, and acid.